All right. Hi, everybody. If you remember on Wednesday, I mentioned that I was going to be out of town for the next few days. I actually came up here to Montana for a friend's wedding. And so we obviously can't meet in person on Friday. But it's important that we stay on track, keep on schedule if we're going to finish everything that we need to talk about this semester. And so I'm going to record the last kind of the second half of the muscle lecture and get this posted for you hopefully later tonight. And I'm also going to take some time to review some of the main concepts that you'll want to make sure that you remember for the exam on Monday. Okay, so remember the exam on Monday is going to cover the bone chapter as well as the muscles. And so that's what we're going to be finishing our discussion of tonight. I thought I'd go ahead and record this outside. It's actually not too cold up here. So anyway, by the way, I hope you feel super sorry for me. I'm actually staying at my grandma's house here. So she's got kind of a neat place by the river. You see a fisherman over there. And enjoying some nice fall colors up here in Montana. So kind of fun to be up here. Let me go ahead and share my PowerPoint with you. I can get this to work here. All right, hopefully everyone can see that okay. It's coming through in the recording. So I'm going to review a couple of the slides that we talked about on Wednesday, and then we'll kind of forge ahead in some new material here. I don't know if this is going to hide any of the words for you, hopefully not. All right, so remember we said that people who are studying muscle physiologists, like to use some terminology that's a little bit unique for muscle tissue. And so if you're thinking in terms of the cellular anatomy of a muscle cell, skeletal muscle cell, a lot of these structures are, cell membrane and various things, endoplasmic reticula. These are structures that we have already seen when we were looking at the, general anatomy of a cell, right? First week or two of class. But they have different terms when you're talking about muscle tissue, right? So we said that if you're talking about a muscle cell, the name for the cell membrane, that plasma membrane, is actually sarcolemma, okay? And then just like other cells in the body, muscle cells have cytoplasm inside of them, but the cytoplasm in this case is called sarcoplasm. So if you see these terms, sarcolemma, sarcoplasma, just remember that those are referring to the cell membrane and the cytoplasm of the cell. And then the endoplasmic reticula in a muscle cell has a special name that's called sarcoplasmic reticulum, and it has a slightly different function in muscle cells than we saw in sort of the general animal cell that we talked about the first week or two of class. Okay, so we'll get back to that here in a couple of slides. This should be a review from Wednesday. All right. A few other structures that you're going to see in a skeletal muscle cell. We've got some, actually, let's start down here at the end, right? I'm going to start at the bottom and work my way up. So the contractile unit, the shortening unit of a muscle cell is called the sarcomere. We're going to see that what that looks like here in a second. If we end up lining sarcomeres up end to end, right? You make a whole chain of sarcomeres attached end to end. What you end up with is called a myofibril. Okay, myofibril is a chain of sarcomeres end to end. Okay, a couple of really important protein filaments that you see inside of a muscle cell, inside of a sarcomere. You've got actin and you've got myosin. Actin and myosin are the two specific proteins that are actually pulling across each other and are responsible for the shortening of the sarcomere in the first place. So we're going to see how that happens here. The entire muscle cell is really long. One muscle cell is actually going to run the entire length of the body of a muscle, right? So if you think about your biceps brachii, right, you've got individual muscle cells that are running from the point of origin up here all the way down and ending at the tendon that is inserting down here on the radius. Okay, so that's, what is that, 10 inches or so? It's a pretty long muscle. You've got even longer muscles than your leg. Okay, so these cells are very long, and because there's such long cells, we sometimes see this term muscle fiber being used. I personally am not crazy about the term fiber because I find it confusing. I feel like it's easy to get that mixed up with, for example, the actin and the myosin, or things like collagen fibers, right? There's a bunch of other fibers that we see in the tissues of the body. So I prefer the term muscle cell, but just know that if you're looking at a slide, or you're looking at your book and it's talking about muscle fibers, remember that a muscle fiber is actually, all that really is a skeletal muscle cell. Okay. By the way, if you have any questions on this lecture, and because you're not able to ask me in person in class, go ahead and shoot me an email and I'll be happy to clear that up for you. Okay. I am trying to keep a close eye on my emails while I'm away. We looked at the sort of the visual illustration here of those different structures in the skeletal muscle cell. Remember, here is that sarcomere. So you have the beginning and end of a sarcomere is marked by this kind of zigzag line. We call that zigzag line a Z line. It's named for that zigzag shape. Okay. And that Z line is really, you can kind of think of that as like a suture. That suture serves as kind of a stationary anchor point at the beginning and the end of the sarcomere. And the contractile filaments, specifically the actin, is going to be anchored to the Z disc, or to the Z line on either end of the sarcomere. Okay, so the Z line is what gives the stationary anchor point for the myofilaments that are going to be pulling against each other. And because they're pulling against that stationary anchor point, as they're pulling toward the middle, that's what causes the sarcomere to shorten. And because your myofibrils are simply one sarcomere connected end to end with other sarcomeres, right? A whole chain of sarcomeres, as all those sarcomeres are shortening, then the entire myofibril is shortening as well. Okay. So this is a myofibril. It's multiple sarcomeres lined up end to end. Okay. You can kind of think of this, it's not membrane bound, but otherwise you could kind of think of this as an organelle within the cell that is the skeletal muscle cell. Okay, so we see a number of myofibrils here in cross section. What do we got? 1, 2, 3, 4, 5, 6, 7. Looks like 8 myofibrils in cross section. The way that the artist has drawn this specific skeletal muscle cell. Okay, so notice we've got a number, multiple myofibrils, all of them housed within a single skeletal muscle cell. Okay, and then just like we see with other animal cells, we see that the outside is surrounded by a plasma membrane, a cell membrane. Remember, that's called a sarcolemma in the case of the skeletal muscle cell. We see mitochondria, just like we saw in other types of cells. We see endoplasmic reticula, let's see, that has a special name here in the muscle cell. It's called a sarcoplasmic reticulum, kind of shown in blue here. Endoplasmic reticula in other cells is involved in sort of modifying and packaging proteins that are built by ribosomes. The function of the sarcoplasmic reticula here is just a little bit different. It's going to store calcium, and it's going to release calcium into the sarcoplasm, into the cytoplasm of the skeletal muscle cell when the skeletal muscle cell wants to contract. So calcium is really critical for contracting of the skeletal muscle cell, and we're going to see how that works in a little bit. But that calcium is stored in sarcoplasmic reticula. Okay. For these channels, kind of think of these as like conduits that allow for the calcium to kind of distribute itself throughout the skeletal muscle cell. They're shown in green here. They're called T tubules. This isn't a structure that we usually test you on in this class, but if you're wondering what those are for, those green structures really are just, you can almost think of them as like drainage pipes that help, you know, distribute the calcium throughout the cell, kind of make sure the calcium can find its way deep into the cell. Okay. You see that skeletal muscle cells are going to have multiple nuclei. We say they're multinucleate. We see a number of these purple colored nuclei in this drawing as well. Okay. So that's everything I've talked about here in the last few minutes. Those are all structures within one individual skeletal muscle cell. Okay. Now, if we bundle together a bunch of individual skeletal muscle cells, what we end up with is a bundle of cells that we call a fascicle. Okay. So this right here would be a fascicle. Here's your myofibril, right? Here's all your sarcomeres lined up end to end to form a myofibril. And then multiple myofibrils are housed within a single skeletal muscle cell. Okay. And then if we have a whole bundle of skeletal muscle cells, that is called a fascicle. Remember, fascia means bundled together. Think of like all the different fibers that are twisted together to form a rope, right? Individual threads or individual fibers in a rope are very weak, right? But if you bundle enough of them together, you can you can pull a vehicle with them. You might go to the shipping yards. I don't know, Houston or we used to go to the shipping yards in Los Angeles, right? These big docks where the huge ships come in, and you'll see an aircraft carrier docked, tied up to the dock. And they use actual ropes. These ropes are, as big around as my legs, very, very strong. So all those fibers together, the word for that, the term for that is fascia. Okay. So if you got multiple muscle cells, multiple muscle fibers bundled together, we call that a fascicle. Okay. And then you take a bunch of those fascicle bundles and you package those together, and now we've zoomed out to the point where you're actually looking at the entire, what we call the belly of the muscle in cross section. Okay. That muscle belly is surrounded by what's called epimysium, right? Epimysium is the connective tissue membrane around the outside of the entire muscle. And that connective tissue is going to narrow to a point, and it's going to form a tendon. And then that tendon is going to be attached to a bone. Okay. Tendons attach muscles to bone. Points of origin, points of insertion, where muscles are attaching to bones, those are held to the bone by tendons. Ligaments are connecting one bone to another bone. All right. So you want to be familiar with these different structures involved in the sort of the organizational levels of skeletal muscle tissue. All right, so let's zoom in, and this should still be a review. We did talk about this at least briefly on Wednesday, but it's a little bit more of an abstract concept. A lot of times, this is the first time you've, this class is the first time you've seen this, so we want to make sure that you're clear on how this is working. Okay, so we're going to take a closer look at what's happening at sort of the molecular level to allow those sarcomeres to shorten, to contract. Okay, so shown in orange here, those are what we call the myosin filaments, or the myosin protein. Okay, and you can notice they've got these kind of these little barbs on them. They're kind of these little heads on the myosin are kind of shaped like little golf club heads. Okay, and those heads, they're actually called actin binding heads, and they're going to reach out, they're going to grab onto the actin and form a bond with the actin protein, and then that sort of golf club shaped head is going to flex. So it's first going to attach. So pretend this is actin. pretend this is myosin. Myosin is going to reach out, it's going to form a bond to the actin, and then it's going to flex. And as it flexes, it's moving that actin along beneath the myosin, okay? So it forms a bridge, and then it flexes and it moves the actin beneath it, okay? In lecture, I said it's a little bit like if you held a centipede, right? I've got a squirming centipede in this hand, and I lower the centipede down onto maybe a pencil. Maybe I'm holding a pencil in my hand, right? Lower the centipede down onto a pencil. Centipede grabs onto that pencil with all of its little legs. And as the legs grab onto the pencil and move against the pencil is going to slide this way, right? Slides beneath the centipede, all right? So that's a sort of kind of a rough description of how these actin binding heads on the myosin are going to reach out, grab onto the actin, and pull the actin across, okay? So the actin binding heads on this side are going to move the actin toward the middle from left to right. Actin binding heads over here on the other side are going to grab onto this actin. The actin shown in red here. Myosin is in yellow. The myosin is going to reach up, grab onto the actin, and pull it toward the center, in this case, from right to left. So, and because the actin are anchored to the Z line, as we're pulling those actin filaments toward the center, the Z lines get pulled together and the entire sarcomere shortens, okay? So you can see that on the bottom here. We've pulled the actin all the way to the middle, okay, as far as we can. And notice that the actin filaments from this side bump into the actin filaments from the actin filaments on the right and the actin filaments on the left. They kind of bump into each other. And there's actually, so there's kind of a maximum contraction. There's a limit to how short you can shorten the sarcomere. At a certain point, the actin filaments on one side are going to interfere with the actin filaments on the other side, okay? And then at that point, the sarcomere is going to stop contracting, okay? All right. So new material here, we're going to talk about the chemistry behind there's actually two different chemical molecules that are involved in facilitating this contraction, the shortening of the sarcomere. Try to move my video out of the way so I'm not hiding any words here for you. All right, so nerves are going to be stimulated by the nervous system. I'm sorry, muscle is going to be stimulated by the nervous system, okay? Nerve impulses, basically electrical impulses, are traveling down nerves, and a nerve comes in and it's going to what we call innervate a skeletal muscle, right? So nerve comes in and it forms a junction with a muscle, okay? And so it's the electrical current, the electrical stimulation coming down that nerve that is going to trigger the series of steps that results in a muscle contracting, okay? So the first step here is that we've got a nerve impulse. We've got an electrical signal, travels down the length of the nerve, and it reaches the end of the nerve where the nerve forms a junction with the muscle cell. And that's how we're going to stimulate muscle contraction, okay? When that nerve impulse, when that electrical impulse arrives at the junction, there's going to be, so this is the end of the neuron, okay? And pretend this is my muscle cell, okay? There's going to be a small gap between the end of my neuron and my muscle cell, okay? Tiny little gap, a tiny little space. On the end of my nerve, there's a bunch of little vesicles. We call them synaptic vesicles. Synaptic vesicle because they are going to release their contents into what's called the synapse, that gap between the neuron and the muscle cell, okay? And those synaptic vesicles are going to release what's called a neurotransmitter. And a neurotransmitter is a molecule that diffuses across that gap. So A neurotransmitter is a signal molecule that diffuses across that synaptic gap, and then it binds to receptors on the muscle side. And we call that neurotransmitter, in this case, it's called acetylcholine. And sometimes you'll see acetylcholine abbreviated ACH. So if you see that abbreviation, just know that we're talking about a neurotransmitter called acetylcholine, okay? So acetylcholine molecules diffuse across that gap between the neuron and the muscle cell, and they bind to receptors on the muscle, okay? Binding of acetylcholine to those receptors triggers a release of calcium ions. Remember, calcium ions are going to be stored in the sarcoplasmic reticulum in the muscle cell, okay? So here's a picture of what that looks like. Here's my neuron, okay? And the neuron comes down and it actually branches so that it forms junctions. It forms synapses with multiple skeletal muscle cells, right? Each of these tubes is a skeletal muscle cell. Okay, so in this picture, we've got 3 skeletal muscle cells, and each of them is innervated by one branch of this specific neuron. Okay, we actually call this a motor unit. A motor unit is one neuron together with all of the muscle cells that are innervated by that single neuron. Okay, so a motor unit has one neuron, and it also includes all of the muscle cells that are innervated by that single neuron. Okay, and you can see these myofibrils in cross section within each of the skeletal muscle cells, right? Okay, so if we zoom in on that, here's the end of one of those neuron branches. Okay, we actually call this an axon terminal. Axon because it's remember the parts of a neuron. We've got the dendrites on one side of the neuron. We have the cell body, and then the projection, the structure of sort of the arm, if you want to think of it, the arm of the neuron that extends out and sends transmissions to the next cell. We call that the axon. So then the end of the axon is called the axon terminal. And it's inside this axon terminal that we have all these little vesicles, all these synaptic vesicles that are filled with that neurotransmitter, that acetylcholine, okay? So remember, a vesicle is able to fuse to a cell membrane, and once it fuses to that cell membrane, it's able to open up and release whatever it's holding on to. And in this case, that means that the synaptic vesicle is going to release acetylcholine. So all these little green balls, those are representing acetylcholine molecules. So the acetylcholine is going to diffuse across the space between my neuron and my muscle cell. And when it binds to these receptors here, that is going to trigger the release of calcium from the sarcoplasmic reticulum. Okay. Be sure to email me if you've got questions on this. I'd even be happy to set up a Zoom meeting with you if especially if maybe there's a group of students that would like to meet with me, run over some of this again in review. We can do that. We've got time Friday afternoon if you want to do that, or Sunday I can meet with you. Okay, so definitely email me if you've got questions that you'd like to get cleared up. If any of this isn't making sense, I'm happy to try to clarify that for you. Okay. All right, so we've got acetylcholine that has diffused across that gap between the neuron and the skeletal muscle cell. We've got sarcoplasmic reticula that are going to release calcium. When the calcium is released, now we're in the sarcoplasm, we're in the cytoplasm of the cell. And the calcium is going to bind to a special protein called troponin. Okay, I realize this slide is getting a little bit wordy, or we're throwing some new terms at you. This is going to make sense when I show you the picture on the next slide. Okay, so we've got calcium getting released from endoplasmic reticula into the sarcoplasm, or sorry, into the sarcoplasm, right, the cytoplasm of the cell. calcium ions are going to bind to troponin. And troponin is a protein that's kind of acting like a hinge. Okay, it's a hinge that holds a couple of fibers together. So we've actually got, remember, we've got myosin, which is the thick fiber, the thick filament in the actin and myosin sort of complex, right? The two proteins that are pulling against each other are actin and myosin, right? It turns out that there are active sites on the myosin that are going to bind calcium ions, okay? And if a calcium ion binds to the active site on a myosin, that is going to, actually, I'm getting ahead of myself. Let's explain that when we're looking at the picture, okay? So sarcoplasmic reticulum dump calcium. Calcium is going to cause movement in a special hinge-shaped protein called troponin. Troponin is going to move a complex called the tropomyosin complex, okay? And it's going to expose binding sites. Actually, the binding sites are on the actin. And when we expose those binding sites, that's going to allow the myosin heads to reach up, lock onto the binding sites on the actin, and then pull the actin across, okay? Let's actually, so for these last few steps here, let's actually look at a picture. I think it's going to be a process that makes a lot more sense if you can see a visual image of what we're talking about here, okay? All right, so refresher here. Here is our myosin shown in yellow. Here's our actin, okay, shown in red. Up here, and this is a little bit frustrating. I'm not sure why the artist chose to do it this way. But up here, these two pictures at the top are actually also showing actin filaments, but they're shown in red. Oh no, that's right. Red on the top, red on the bottom. All right. So actin is in red in both cases, and it's the myosin that is in yellow. Good. Okay. The other book I used to use, they swapped the colors on us. All right. So actin filaments, we call those the thin filaments. These are the filaments that are, remember, they're anchored onto the Z lines, onto the beginning and the end. At the beginning and the end of our sarcomere, we have actin filaments. Okay, so if this is a Z line, my actin is going to be sort of tethered to this Z line. Okay, so the actin is held in place because it's attached to the Z discs, right? The actin filaments have these little binding sites. You kind of see them shown. You see these little dark areas here. Those are the binding sites where the myosin heads want to come up and form a link so that they can grab onto the actin and pull the actin across. Okay. But notice that the binding sites are blocked by this screen. We call this tropomyosin. Okay. So the actin filament is kind of, it's a couple of different threads, actually, protein threads wrapped around each other. And then within that groove, within that twisting groove, are the binding sites, okay? But when the muscle is relaxed, those binding sites are actually blocked by this green protein called tropomyosin, okay? And it's held in place by these hinges, these protein hinges that are shown in blue. That's the troponin, okay? So we kind of have three proteins here in this picture. We've got actin that's shown in red. We've got tropomyosin, which is shown in green, and it's blocking the active sites, the binding sites on the actin. And then we have these hinges, these protein hinges that are made of troponin that are holding the tropomyosin in place. All right. So the key here to understand is that myosin has these binding heads that really would like nothing more than to be able to reach up, grab onto the actin, and slide the actin across. Okay, but they're blocked from forming that that bond because the binding sites are hidden. They're obscured behind this tropomyosin. Okay. So if we want the muscle to contract, we need to find a way to move that tropomyosin out of the way so that the binding sites are exposed and the myosin heads can come up and form that link. Okay. So it turns out that the way that we do that is that we need to have calcium ions bind to the troponin. Turns out that you actually need 4 calcium ions to bind to each troponin hinge in order to get that hinge to basically pull away and pull the troponin out of that binding site. Okay. All right. Once we have removed the troponin from blocking the binding site there, then the myosin head can pull up and attach to that binding site and pull the actin across itself. Okay, so you have this cycle that's happening, right? I'm actually going to start over here with step 4. This is a cycle, so you can actually start at any step that you want. To me, it makes more sense if I start with what's called step 4 in this picture. Okay, this is a step that requires energy. You need to spend energy in order to make that that binding head reach up and bind onto actin. So notice we've got ATP shown here. We spend ATP to have that binding head reach up and attach to the binding site on the actin. Okay, so we spend ATP. We split the ATP to release that energy. Okay, and that actually happens right before the myosin head actually attaches and binds to the actin. Okay, so what's labeled step 2 here is that we actually form a cross bridge, a cross link between the myosin head and the binding site on the actin. Okay. The last step is that binding head is going to flex, and as it flexes, it pulls against the actin. It's going to pull the actin in the opposite direction. Okay, so the myosin is kind of moving. In this picture, the myosin is going to be moving left to right, and the actin is going to be moving right to left. Okay. And remember, over here somewhere, you're going to have this actin being tethered to a Z line. And so if we're pulling against that Z line, we're going to shorten the sarcomere from that side. Okay. Then the cycle just repeats itself. Okay. We spend ATP. We move our myosin head up into position so that it's ready to form a cross bridge. We form that link. We bind the myosin head to the active site on the binding site on the actin, and then we flex that head. And as the head is flexing, it's pulling the actin across itself. Okay, and this is happening at each of those little binding heads. We've got lots and lots of myosin heads on each little thread of myosin has its own binding head. So these are like all of the multiple little legs on a centipede. Each of those is independently reaching up, forming links with the actin and pulling the actin across itself. Okay. No, there's a lot of steps there, and that sounds a little bit complicated. For the test, what you really need to know is that there are 2 key chemicals that are involved in contraction, and those are acetylcholine, that's the neurotransmitter that's once the electrical impulse comes down the neuron, reaches the axon terminal, causes the synaptic vesicles to release acetylcholine. The acetylcholine neurotransmitter molecules diffuse across that gap. They bind onto acetylcholine receptors on the muscle side. When the acetylcholine receptors are when they bind a neurotransmitter molecule, then that triggers the sarcoplasmic reticula to release their calcium. Calcium is released into the sarcoplasm, and the calcium then is going to go out. It's going to find troponin hinges to attach to, and that's going to allow the troponin to pull back As the troponin pulls back, it's going to move the tropomyosin out of the way so that it's no longer blocking those binding sites. And once those binding sites are exposed, we're able to spend ATP to form a link between the, spend the ATP to first move the myosin head up into position, and then form a cross-link with the actin, and then finally flex the myosin head and allow it to pull the actin across itself, okay? Acetylcholine is the neurotransmitter, and it results in the release of calcium. Acetylcholine, calcium, those are the two chemicals that are really important for allowing contraction of skeletal muscle, okay? All right, the energy for that process is provided by ATP, okay? Just kind of an interesting side note, after a person passes away, same thing happens in an animal, right? The cells actually in those tissues continue to live for a certain amount of time. Obviously, if the organism's passed away, they're no longer breathing, so there's no oxygen coming in. There's very limited cell respiration that can be happening. But even in the absence of oxygen, remember that fermentation can be happening, okay? And fermentation means that there is still some production of ATP that's being produced, and we're not producing, 36 molecules of ATP for every molecule of glucose, but we are producing at least a couple of molecules of ATP in fermentation. And so there is still some ATP available, and muscle cells can continue to contract even after an individual is no longer living, okay? At a certain point, though, you're going to run out of ATP, and some of the myosin heads are still going to be attached to the actin, right? And because we need to spend ATP not only to get the myosin heads to attach to the actin in the first place and to flex, but also it actually also requires ATP to cause them to release. And so if you've used up all of your ATP, the organism's passed away, you're no longer bringing in oxygen, there's no cell respiration happening, you can't make more ATP. So you're going to run out of ATP eventually. And any myosin heads that are still bound to actin, when you run out of ATP, they're going to stay attached to the actin. And so the muscles are continuing to contract after death. And this is that term that you might have heard if you watch, crime detective shows on TV, right? Rigor mortis, right? For a certain period of time, maybe a couple of hours or so after death, the body is really tense and rigid, right? That's because those actin heads are still bound to the ATP, bound to the actin proteins, and they can't let go without ATP. At a certain point, as decomposition starts to happen, those actin heads are going to let go of their the myosin heads are going to let go of the actin, and the muscles are going to relax, okay? All right, some terms here. A couple of these I've mentioned already. Remember that a motor unit is a nerve, and all of the muscle cells that it stimulates, okay? So it could be one nerve and 10 muscle cells. It could be one nerve and 100 muscle cells. Motor units come in different sizes, but it's always going to be one single neuron and all of the individual muscle cells that are innervated by that particular neuron, okay? If you have a nerve impulse reach an axon terminal, release acetylcholine, and trigger contraction of a cell, we call that a muscle twitch. One single contraction. It's very brief, okay? A muscle twitch is just a single nerve impulse resulting in a single release of calcium ions from the sarcoplasmic reticulum and one single shortening of all of these sarcomeres, all of the myofibrils in that particular muscle cell. Okay. Now, if we have multiple twitches happening in rapid succession, what we can have is we have what's called summation. Okay, summation is the sum of what you get when you add a whole bunch of twitches together. It turns out that when you have a nerve impulse come and trigger release of calcium into the sarcoplasm, right, and that calcium causes shortening of the sarcomeres. If you want to allow those sarcomeres to return to their original size, the way that you end muscle contraction is you actually have to pump those calcium ions back into the sarcoplasmic reticulum. Okay, as long as there are calcium ions bound to the troponin are going to continue to be pulled out of the way and exposing the binding sites on the actin, so that those myosin heads can continue to reach up and grab onto the actin and pull it across. Okay, so if you want the muscle to relax, you have to actively pump the calcium ions out of the cytoplasm of the cell into the sarcoplasmic reticulum. Okay, and that takes a little bit of time. So a muscle cell can't just relax instantaneously. Because it takes, a moment to pump that calcium back out, if we send a second nerve impulse right away, so one after another, we can cause those muscle twitches to add up. Okay, so if you add multiple twitches together in rapid succession, what you end up with is summation. Okay, and so summation is a more extended muscle contraction. It's not just a fraction of a second. It might be 1 second, it might be 30 seconds, it might be a minute, right? As long as you're contracting that cell, what you're doing is you're continuing to send nerve impulses to that muscle cell, and you're continuing to tell it to dump calcium into the cytoplasm so that we can continue to have the binding sites exposed there for the myosin to bind onto the binding sites on the actin molecules. Okay. Tetanus is the term that you end up with if you have like permanent contraction of the muscles. And sometimes this can be caused by, we most often think of tetanus as something that's caused by actually a bacterium that lives in the soil. A lot of times we associate this with like rusty metal, right? So if you cut yourself on a piece of rusty metal, or if you step on a rusty nail, sometimes you introduce that microorganism into your body, and it actually attacks the neuromuscular tissues. It causes permanent contraction of your muscles. Obviously not something that you really want to happen. Tetany is maximal sustained muscle contraction, and is usually not something that you can voluntarily do. It's usually, if you're seeing true tetany, that's usually only showing up in really unusual circumstances, such as if you've got a microbe that's attacking the neuromuscular junction. Okay. Tone. Okay. You always are going to have partial contraction of muscles in your body. None of your muscles are ever going to be fully relaxed at a given point in time. They're always going to have a certain amount of partial contraction that's happening, and it kind of it's what allows your muscles to have some shape to them, right? Come on, there we go. So here's what I was talking about in terms of, this is what a single contraction of a muscle cell looks like. Okay, this is a twitch. So you've got a shortening of the sarcomeres, the myofibrils, all of the sarcomeres joined end to end. Those are shortening. They reach maximal contraction, and then they release, and they go back to their original length. That single muscle contraction is called a muscle twitch. But notice that it takes a certain amount of time for us to be able to pump that calcium out of the sarcoplasm back into the sarcoplasmic reticulum. So we have a moment of, it takes a little bit for that muscle to relax. And so if another nerve impulse comes before that muscle cell is able to fully relax, then you end up adding twitches onto each other, okay? Notice that these peaks don't ever reach their baseline before the next peak comes along. And as we add those peaks together in more and more rapid succession, we have a more sustained muscle contraction. And here's that tetanus term here, the maximal muscle contraction, that you almost never see in a normal physiological circumstance, okay? All right. All right, so where are we getting the energy for muscle contraction? Well, it turns out that we have really four types of organic molecules that store a lot of energy in their chemical bonds, and we can use all four of those as sources of energy. In the muscle tissue itself, you have glycogen and you have triglycerides, okay? So remember that glycogen is that molecule that so after a meal, we've got lots of glucose, right? We've digested our food, we've broken that food down into glucose. We've got glucose floating around in the blood. In the body, remember that, physiologically, we're all hunter-gatherers, right? Our bodies don't like to let energy go to waste. And so when our body cashes that paycheck, right, when it's got that glucose in the bloodstream, it's going to use that glucose for all of its immediate energy needs. But then if there's any glucose left over, it's going to store that glucose. And the way that it stores that glucose is that it adds those individual glucose molecules onto a huge storage molecule that we call glycogen. Glycogen is just a complex of tons and tons of individual glucose molecules all bound together, okay? So glycogen is an energy storage molecule. It's in your muscle tissue, okay? And it's one of the first energy sources that your body accesses when it needs energy, okay? The other type of energy storage molecule in your muscle is triglycerides, okay? So remember, if you break down fat, you end up with triglycerides and fatty acids, okay? So triglycerides are kind of a part of fat, and glycogen and triglycerides are both stored in your muscle tissue. In the blood, circulating throughout your body in the bloodstream, you've got glucose, so simple sugar, and you have fatty acids circulating in the blood as well. When you exercise, if you go to the gym and you start, running on a treadmill, for the first, I would say 20 or 30 minutes of, fairly rigorous exercise, your body is preferentially going to be accessing the glycogen that's stored in your muscle, okay? Going to be accessing those glycogen reserves in your muscle. You can see that in the green bar here in this figure. It's not going to be using much of the glucose in your bloodstream, okay? That's the orange bar here. And it is also going to be using some of the triglycerides in your muscle, some of the fatty acids that are floating around in your blood, okay? But notice the longer you exercise, the less and less your body is using the glycogen in your muscle for energy, because you're actually depleting the glycogen in your muscle. And you actually start, as you exercise longer, and notice this is quite a while, right? Most of us aren't exercising for 4 hours at a time, unless you're a marathon runner or you're going for hikes out in the woods. Maybe some of you enjoy road cycling, right? Maybe you have a road bike that you take out and you go for 4-hour rides. Most of us are doing good if we exercise for an hour at a time. But anyway, if you push your body and you're going for 4 hours, notice that you actually kind of deplete your glycogen reserves, okay? At the 4-hour mark, we're not using glycogen as a source of energy. Instead, what you see increasing the longer you exercise is you come to rely more and more on fatty acids and glucose. You start to rely more on the energy molecules that are circulating in the blood, and not so much on the triglycerides and the glycogen in the muscle. Okay. So for the test, I'd like you to be able to name 4 different molecules that are sources of energy: glycogen, triglycerides, glucose, and fatty acids. And I'd like you to know that the glycogen and the triglycerides are stored in muscle tissue, whereas glucose and fatty acids are circulating in the blood. All right, I'm actually going to skip over the next couple of slides. This gets into some muscle physiology that I just am not super interested in you learning for this class. We're throwing a lot of information at you, and you've got enough that you need to worry about for Monday's test. So don't worry about the creatinine phosphate pathway. Cellular respiration, you do know the basics of what cellular respiration is, right? That's how we're using glucose, for example, and breaking that glucose down and producing ATP. And fermentation is basically cell respiration that's happening in the absence of oxygen, right? So this would be fermentation is an anaerobic process, right? Whereas cell respiration is an aerobic process. It uses oxygen, okay? Both both aerobic cellular respiration and fermentation are producing ATP. The difference is that aerobic respiration is producing way more ATP than fermentation is, okay? So again, let's not worry about the creatinine phosphate process here. I'm just going to skip this slide. Okay, don't worry about this slide. All right, in terms of our muscle tissue, we actually have 2 different, well, there's actually 3 different types of muscle fibers. We're going to talk about 2 of them here. You've got fast twitch muscle fibers, and you've got slow twitch muscle fibers. Okay, and they have different functions, right? You have different uses for those 2 types of muscle fibers. Okay, we're not going to worry about creatinine phosphate. Not going to worry too much about fermentation. But what I do need you to know about fast twitch fibers is that it's fast twitch fibers that you're using for strength. Okay, we use these for when you're lifting weights, right? When you're really kind of wanting to use all of the potential force that your muscles can produce, you're using fast twitch fibers. Okay, if you look at fast twitch tissue under the microscope, right? If you're doing a dissection, whatever, they're going to appear pretty light in color. Okay. Part of the reason for that is they don't have a lot of mitochondria. Okay. And there's very little myoglobin. Okay. And few blood, they're not very highly vascular, right? Fewer blood vessels. This is less metabolically active tissue. Can produce a lot of force, but it's not very vascular, right? There's not much myoglobin there, okay? Not very many mitochondria, right? Slow twitch fibers, they are not so much for strength, but they're for endurance, okay? They are going to be a little bit darker in color. They've got lots and lots of mitochondria, very mitochondria rich, lots and lots of myoglobin, and they're very, very vascular. Lots of blood supply in slow twitch fibers. And your muscles are actually mixtures of both slow and fast twitch fibers. Some of your muscle groups have, predominantly fast twitch. Some of your muscle groups have predominantly slow twitch muscle fibers, okay? But most of the muscles in your body are going to be a mix of the 2, okay? You can think of, muscles that you're using all the time, right? Like, what would be a good example? Muscles in your legs that are helping you walk, right? We use these all the time, and you've got pretty good endurance, right? The postural muscles, right? Your abdominal muscles of your torso, whether or not you realize it, they're working all the time just to keep you upright, just to keep you, standing up, sitting up, and not, slumping over and, up in a pile on the floor, right? So you're using your postural muscles all the time. Those would be a good example of, higher endurance, slow twitch fibers. Okay. They're not very strong, right? But they've got endurance. When you're lifting weights, you're going to be using your fast twitch fibers. These are the lighter colored ones here. But they fatigue pretty quickly, right? If you watch powerlifting, right? They might be able to lift an incredible weight 2 or 3 times. But if you ask a weightlifter to lift at their maximum weight 4, 5, 6 times, they're going to get tired, right? Those muscles are going to fatigue pretty quickly. Okay. Whereas slow twitch fibers, you're able to use those for a much longer period of time before they start to fatigue and get tired. Okay. So as we start to wrap up, we'll look at some of the health implications that you should be aware of in terms of the muscle system, right? When you're exercising, the more we exercise our muscles, obviously, we all know that we're increasing the strength of our muscles, right? When you lift weights, if you lift weights consistently, you're going to find that your strength increases and you're able to, lift consistently, increasing, consistently increasing amounts of weight, right? So you're improving the strength of your muscle. You also improve the endurance of your muscle and the flexibility, right? So those are all good things. Those are all benefits of exercising. We're also improving our cardiorespiratory endurance, cardiorespiratory health, right? You're exercising your heart, exercising your lungs, all of that is good. Your heart and lungs get into good shape. Just like you can strengthen the strength, the endurance of other muscles in your body, you can also strengthen and improve the endurance of your heart and your lungs, okay? Diaphragm, right? We can improve the ratio of HDL to LDL, bad cholesterol compared to good cholesterol in the body, good lipids, bad lipids, right? HDL is the good lipid that you want to see in your body. LDL is we say lousy lipids and happy lipids, right? HDL is good, LDL is bad. You want to see a higher ratio of HDL to LDL, right? HDL, by the way, is going to transport lipids to be burned, right? LDL transports lipids to be stored. And so you do want to store some lipids, you just don't want to store a lot of them. You'd rather see lots of lipids getting sent to be metabolized and burned off. So that's why we like to see higher levels of HDL compared to LDL, okay? And that ratio improves as we exercise. We also see healthier ratios of protein to fat. The more that we exercise, right? You're burning fat as you exercise, you're building protein, building muscles, okay? People who exercise regularly are going to have lower risk of certain types of cancer, colon cancer, breast cancer, cervical cancer, uterine, ovarian, okay? Notice a lot of these are, only, cancers that women have to worry about. But even guys obviously are going to be glad to be able to reduce our risk of colon cancer, right? Colon cancer is one of those that if you catch it early, there's a pretty good prognosis, right? But a lot of us don't catch colon cancer until it's more advanced, right? And it can be pretty serious, right? Breast cancer, guys can get breast cancer. I'm not sure if I mentioned that before, but breast cancer is not just something that women get. Guys need to worry about that as well. And so if we can, anything that we can do to reduce our odds of getting these types of cancer, going to be a good thing. Exercise is going to help our bones become more dense, right? As muscle is pulling against bone, it's stressing the bone at that point where the tendon inserts on the bone. And as we stress the surface of the bone, all of your osteoblasts in that part of the bone are going to be busy building new bone material there and actually reinforcing that bone where the stress is occurring. And so you're actually remodeling and strengthening your bone material as you exercise. Also, impact exercise is really important for bone health. It turns out that as you are compacting bones, so think about when you're running, right? Every time you land on your feet, you're producing impact forces across the long bones in your legs, right? Tibia, fibula, and the femur. You're impacting those bones, and those vertical impact forces are actually prompting the osteoblasts in those bones to get busy reinforcing and building more, laying down more bone material. And so your bones strengthen themselves and become more dense with impact sports. We actually find that it's possible, it's maybe not common, but there are situations where an individual might be an amazing cardiovascular health, right? Maybe they're a cyclist and they put hundreds of miles on their bike every week, right? Hours and hours of exercise on the bike. Or maybe you've got a swimmer that spends hours in the pool every week. So they're in excellent health. But if you're only doing low impact exercise like swimming and bike riding, and you're not subjecting your bones to impact, right? We actually see a decrease in the density of your bones. And so it is important to involve at least a certain amount of impact activity every day to stimulate the bones remodeling and reinforcing themselves and staying nice and dense. Okay. And then obviously exercise also is going to release endorphins and improve your mood. It's a great stress reliever. Okay, lots and lots of benefits to regular exercise. Right, some things that can go wrong in muscle tissue: spasms, seizures, cramps, strains, sprains. These are all terms we've heard of before. Spasms are when your muscles start twitching without you telling them to. A lot of times this can have to do with maybe a mineral imbalance. Sometimes things like, not having enough potassium in your diet can cause muscle spasms. Seizures, a lot of times seizures are more of a neurological issue where those nerves are sending impulses to the muscles when they're not supposed to be. And so causing, unwanted contractions and spasms of the, well, I shouldn't say spasms, unwanted contractions of the muscles, causing a person to seize. Cramps, this is where your muscles kind of contract and firm up, and it can be pretty painful here. This again can have to do with a lot of times with mineral imbalances. If you're not getting all the vitamins that you need, those types of things can contribute to cramps. Also, hydration can be an important one. If you are dehydrated and you're really exerting yourself hard. I remember I was in high school. We went on a band trip, and we one of the hotels we were staying at had a swimming pool, and a bunch of us were playing water polo in the swimming pool, and we weren't drinking enough water. We weren't taking in enough electrolytes, and we're swimming hard, running hard in the water, jumping, right? So you're really working those leg muscles a lot, but you're not properly hydrating, and you're not getting all your electrolytes like you should. And I remember cramping up really, really bad, super painful cramps in my calf muscles. Really something that you want to avoid if you can. Cramps can be very painful. Strains are where you basically pull your muscles too hard. You put too much force, pulling forces on those muscles. You can actually cause stretching or partial tearing of the muscle, right? So that's a muscle strain. A sprain is where you twist a joint a way that it's not supposed to twist. And this is more of a soft tissue injury, right? Ligaments, tendons can be damaged. A lot of times there'll be bruising. If you sprain your ankle really bad, a lot of times it will bruise because you've damaged some of the blood vessels around that joint, right? And it's painful too, because there's a lot of nerves in there as well. Okay. Some diseases that can affect the muscle system, right? You might have heard of fibromyalgia. Fibromyalgia is a disease we don't actually understand an awful lot about, but basically what's happening with fibromyalgia is that you experience really achy muscles. It's just chronic aching of the muscles, and we aren't exactly sure what causes it, but it's not fun. It's something you rather not have to live with if you don't have to. Muscular dystrophy, muscular dystrophy, can't talk. This is a genetic disorder. It's actually a group of related genetic disorders where you've got progressive degeneration of the muscle tissue, weakening of the muscles. Myasthenia gravis is an autoimmune disease. Autoimmune means that your body's own immune system is attacking attacking the body's own tissues, and in this case, the immune system is attacking the receptors, the acetylcholine receptors on the muscle cells. So remember that nerve releases synaptic vesicles containing the neurotransmitter acetylcholine across that gap. So if you've got a patient that's got myasthenia gravis, the nerve is releasing the acetylcholine. The acetylcholine is diffusing across the gap like it's supposed to, but the immune system has been attacking the proteins, the transmitter receptors on the muscle side. And so if you don't have the receptors for the acetylcholine, you can release as much acetylcholine as you want into that gap, but it's not going to cause the muscles to contract properly because you don't have enough receptor proteins on the muscle. Okay. Amyotrophic lateral sclerosis. We typically just abbreviate this as ALS, or you might have heard it called Lou Gehrig's disease, right? This is where the motor neurons actually start to break apart and degenerate. As those motor neurons degenerate, you're not able to, you're not able to send the nerve impulses to the muscles like you want. And so the person progressively loses more and more control over their muscles. It's really a sad thing. And it usually happens pretty quick. Most types of Lou Gehrig's disease progress fairly quickly over a few months. Okay. All right, and then lastly here, I'll just wrap up by talking about steroids. I will say that this is going to be one of the options, short answer options on the test. You might want to make yourself familiar with steroids, particularly in sports. Athletes sometimes end up using what are called anabolic steroids. So you think about this term anabolic. If you remember, there's a pair of terms, anabolic and catabolic. And this kind of goes back to like the very first week or two of the semester when we were talking about organic molecules. The process of building a molecule is called anabolism. And if we're breaking molecules, like when we break ATP to release energy, that process of breaking a molecule is called catabolic. So catabolic is breaking down a molecule. Anabolic is building a new molecule. And so what anabolic steroids are doing is they're actually triggering your body to build more protein. Okay, you're building more muscle. If you're taking an anabolic steroid, you're building more muscle. Baseball fans might have heard of Sammy Sosa, right? And Toby, what was his name? Was it Toby Maguire, right? With the Cardinals, I think. Anyway, every once in a while we have an athlete who decides that they're going to try to shoot up with anabolic steroids to increase muscle mass, and they gain a lot of strength that way, right? There's some really important side effects here. So not only are you cheating by using these steroids to bulk up, right, instead of doing it the old-fashioned way, lifting weights and exercising like the rest of the athletes are. So not only are you cheating, but you're also risking some really important side effects, right? So anabolic steroids tend to increase blood pressure. They can increase kidney problems that lead to jaundice. Jaundice is where you have a yellowing of the skin because the urinary system isn't able to, the kidneys aren't able to filter and eliminate metabolic wastes like they're supposed to. Acne, maybe not a huge deal, although people who are using anabolic steroids extensively can develop, pretty severe cases of acne. Okay, you're also increasing your risk of cancer, testicular cancer, ovarian cancer. There might be other types of cancer that are at increased risk due to steroid use. Okay, you'll also see some effects on the reproductive system. So you'll see, especially in guys, the risk of impotence is going to be much greater if you're using these drugs, and actually shrinking of the genitals as well, shrinking of the testes in this case. Okay. Increased aggressiveness, violent mood swings. One of the steroids that's often injected is testosterone. And so if you're using lots and lots of testosterone, testosterone is actually associated with risk-taking, with aggression, right? In addition to increased muscle mass and those types of things. There are some other things that testosterone can do that are much less desirable, right? And so sports affiliations have, largely outlawed the use, almost universally outlawed the use of anabolic steroids because they're not worth the risk, right? Because they have important health implications as well as, not being sporting, right? It's kind of taking the easy way out to achieve the results that you're looking for. Okay. All right, so we've got a nice summary slide here for you. Remember that we've got 3 types of muscle tissue. We'd like you to be familiar with skeletal muscle, smooth muscle, and cardiac muscle, right? If I were to show you a picture of those types of muscle cells, I'd like you to, or a description of those different categories of muscle, I'd like you to be able to identify those. I'd like you to be able to, talk intelligently about the, give an informed description of the functions of the skeletal muscles, specifically the skeletal muscle system, right? Muscular system being mainly this, we're focusing on the skeletal muscle in this chapter. There are a short list of muscles that we need you to learn by name. That list is available on Canvas. When I do the review video and get that posted, I'll go over some of those with you. Remember that muscles are often named for their location or their shape, or maybe the exact action that they're doing, right? Whether they're flexing or extending or abducting or adducting, right? The name oftentimes will give you really important clues about where the muscle is located, what action it's doing, or the shape of the muscle. Okay. I'd like you to be familiar with the process of how contraction in muscles happens, why ATP is important. Also, not on this list, but super important to remember that it's going to be acetylcholine and calcium that are the 2 chemicals that facilitate contraction of muscle. Remember, rigor mortis is that sort of phenomenon that we see that is where after an organism dies, you still see muscle tension in the tissues because the myosin heads are bound to the actin still, and you have to spend ATP to release the myosin from the actin. And so if you run out of ATP, any myosin heads that are still bound to actin are going to stay that way. So the body can be stiff and rigid after for a couple hours at least after death. 2 types of muscle fibers. We've got our slow twitch and our fast twitch. Fast twitch, remember, is lighter in color. Slow twitch is darker in color. Slow twitch has more endurance. Fast twitch has greater strength, right? Remember the benefits of exercise, some of the common muscle disorders, okay, muscle diseases, and kind of know some of the basics of anabolic steroids. All right, so that is kind of a wrap up of the muscle PowerPoint. I'm going to stop sharing this See, how long have I been going? I think I ran a little long. Sorry about that. Sorry if this video is a little bit longer than 50 minutes, but it was important for us to get through that. I am going to go ahead and record a separate a review video. I hope to get that posted maybe tomorrow for you so that you can take advantage of that over the weekend as you are getting ready for the test. Please just don't be shy about reaching out to me and letting me know if you've got any questions on this material. I can definitely try to clear that up for you. Want to do everything that we can to help you feel prepared for this next test and ready to just really do well on it. All right. So thanks so much for your attention. And we will see you in class Monday for exam number, I think we're on exam number 4 now. Okay. Remember, the exam is going to be over chapters 12 and 13, the bone skeletal system and the muscle system. Okay. Thanks a lot, everybody.